Namaskar, welcome to this session. My name is Lalita Ledwani and I am working as a professor of chemistry and director School of Basic Sciences at Manipal University, Jaipur. Fecal sludge term is given to undigested or partially digested slurry or solids resulting from storage or treatment of black water or excreta. Today, we shall learn about physical-chemical constituents of fecal sludge. I shall give you an overview of commonly measured constituents for characterization of fecal sludge. Excreta contains nutrients that originate from food consumption. Of the total nitrogen, phosphorus and potassium that is consumed, 10 to 20% of nitrogen, 20 to 50% of phosphorus and 10 to 20% potassium is excreted in feces. 80 to 90% of nitrogen, 50 to 65% of phosphorus and 50 to 80% of potassium in the urine. Nitrogen emitted in fecal sludge is responsible for the production of ammonia in the process called deamination. The majority of ammonia in raw fecal sludge comes from the urine, and the nitrogen content in feces is about 20% as ammonia, 17% of organic nitrogen in the living cells, and remaining as organic nitrogen, for example, protein and nucleic acid. Nitrogen content in fecal sludge is typically quite high. In general, it is 10 to 100 times of the concentration in domestic wastewater. Nitrogen is present in different combinations such as ammonium, ammonia, nitrate, nitrite and in organic forms, for example, amino acids and amines. These combinations are dependent upon factors such as pH, length of storage, presence of oxygen and the types of fecal sludge itself. Total Geldar's nitrogen, that is TNK, is the sum of organic nitrogen and ammonia. Nitrogen is estimated by Geldar's method and flow injection analysis method. Nitrate and nitrite can be determined by ion chromatography, capillary ion electrophoresis, cadmium reduction, hydrogen reduction, cadmium reduction flow injection or UV spectrometric method. There are also commercial available kits for these analyses. Total nitrogen can be determined by oxidative digestion to nitrate followed by quantification of the nitrate by sum of total Geldar's nitrogen and nitrate-nitrite results. Phosphorus is another component. The concentration of phosphorus is also an important parameter to consider, as total phosphorus concentration in fecal sludge is quite high. For example, it is 2 to 250 times of the concentration in domestic wastewater. Phosphorus in fecal sludge is present as phosphate. The efficiency of phosphorus removal in various treatment processes is based on factors such as absorption, precipitation, complexion, sedimentation, mineralization, pH, plant uptake in planted drying beds and redox potential. Phosphate can be determined calorimetrically to determine reactive phosphorus or following hydrolysis or digestion to quantify total phosphorus, including particulate and organic fractions. Next component is pH, which is referred as negative logarithm of hydrogen ion concentration. It is an important quality parameter for fecal sludge characterization. Measurement of pH is essential for the understanding of water chemistry processes such as acid-base chemistry, alkalinity, neutralization, biological stabilization, precipitation, coagulation, disinfection and corrosion control. The pH of fecal sludge from septic tanks is normally present in the range of 6.5 to 8, but it can vary greatly from 1.5 to 12.5. A pH outside the range of 6 to 9 indicates an upset in the biological process that inhibits anaerobic digestion and methane production. This could result from a change in hydraulic loading and the presence of toxic substances, a large increase in organic loading, or that the systems are receiving industrial or commercial wastewater. The pH can be measured with electrodes, by pH meters and by pH papers. Another important parameter used for the estimation of fecal sludge is total solid contents. The total solid concentration of fecal sludge comes from variety of organic, which are volatile, and inorganic matters. It is comprised of total solids, fraction of volatile or fixed solids, and suspended or dissolved solids at a specific temperature in the range of 103 to 105 degrees Celsius. The total solid are quantified as the residue remaining after a waste sample remaining after 24 hours of drying in an oven at 103 to 105 degrees Celsius. The total volatile solids are the fraction that are ignited and burnt off at temperature of 500 degrees Celsius. This fraction is also considered to be an organic portion. The fixed solids are the amount remaining after ignition and are generally considered to be the inorganic portion. The total solids values are important as they are used to design and dimension of fecal sludge treatment technologies such as planted and unplanted drying beds. The suspended solid fraction of fecal sludge are defined as solids that are not able to pass through a filter, while the solids that do pass through are termed as dissolved solids. The oxygen demand of fecal sludge is an important parameter to monitor, as the discharge of fecal sludge into an environment can deplete or decrease the oxygen content of water bodies, resulting in the possible death of aquatic fauna. The oxygen demand reduced through stabilization and can be achieved by aerobic and anaerobic treatment. Biological oxygen demand, that is BOD, is a measure of oxygen used by microorganisms to degrade organic matter. The standard method for detecting BOD involves incubation at 20 degrees Celsius for 5 days. Wastewater is considered to be weak, medium, strong, very strong respectively, as at BOD 200, 350, 500 and 750 respectively. Fecal sludge typically has much higher BOD than that of strong wastewater. Non-carbonous materials can also consume oxygen, for example, the oxidation of ammonia to nitrate, which can increase the reported biological oxygen demand value if not taken into an account. To prevent this, nitrification can be inhibited through the addition of chemicals. The particle distribution has an effect as smaller or more soluble particles have faster biological oxygen demand reaction rate coefficients. Other factors that can be taken into an account are sample variability, including filtration, dilution, and sample methodologies. BOD only represents biodegradable organics, whereas COD represents oxygen equivalent of organic matter that can be oxidized chemically with dichromate, a powerful chemical oxidant. COD concentration will always be higher than BOD for a number of reasons, including complex organic molecules like lignin are resistant to biodegradation, being oxidized by COD. Another reason, some inorganic substances also being oxidized by COD. Next one is inhibition of bacteria in the BOD test. COD is determined in the laboratory with an open or closed reflex method and also through commercial kits. The ratio of BOD to COD can also be used as an indicator of the relative biodegradability of the organic matter in different waste streams. Another important component is oil and grease. Fat, oils and grease in fecal sludge comes from wide variety of sources including lard, meats, seeds and kerosene and lubricating oil. Oil grease content is important to consider because it can reduce microbial degradation due to reduced solubility, increase the scum layer in settling tank. It can also cause maintenance problem and create film in surface water if discharged. Extraction in solvents can be used to determine concentration of oils and grease. It is reported as total oil and grease soluble in specific solvents that is used. Grit and sands are important to consider in the treatment of fecal sludge as their present influences the required and filling rates of tanks used for storage and treatment. It can increase the frequency of clogging in pipes and pumps. In areas where sand is pervasive in the fecal sludge, it is a significant treatment and design concern. In order to reduce the sand and grit content, sand traps should be installed at the entry points of pipes and sinks. Concentration of sands can be determined by first drying the samples at 105 degrees Celsius in an oven, then at 550 degrees Celsius to obtain total fixed solids. The ashes are then treated with hot mixture of nitric acid and hydrochloric acid. The amount of sand is obtained after filtration following calcination at 1000 degrees Celsius. Exposure of untreated fecal sludge should always be considered as a pathogenic risk. Adequate reduction in pathogens need to be determined based on intended use or disposal option for treated sludge and liquid effluents. Some common pathogens of concern that may be excreted in feces is presented here. Indicators play important role when measuring the treatment efficiency of fecal sludge treatment process. It is too expensive and labor intensive to measure all types of pathogens. Instead, it is common practice to select indicators for pathogenic activity which are measured to provide an indication for the level of pathogens removal during treatment. These indicators can either pathogenic or non-pathogenic, but the organism need to be carefully selected in order to provide adequate information on the inactivation of pathogens. The following requirements should be met when selecting an indicator. Be exclusive of fecal origin. Be in number greater than those of pathogens of concern. Have a removal then mimic and is close to that of pathogens of concern. Be simple, inexpensive, accurate and reliable to measure. In addition, indicator organisms should have ability to survive bacteriophage as an indicator of viruses. Other indicators of fecal contamination that have been used in pollution control and pathogens die-off studies include fecal streptococci, Clostridium perfringens, bacteriocytes, enterococci, and bifidobacterium. Coliform bacteria are bacteria that populate the intestinal tract and are pervasive in feces. Their presence in the environment is therefore used as an indicator in fecal contamination. E. coli is the target organism that has traditionally been used to identify fecal contamination in the environment. However, there are complicating factors such as bacteria from genus Escherichia that can grow in the environment, and the test is therefore not 100% indicative of fecal contamination. Tests have been developed to enable the quantification of total coliform, fecal coliform and E. coli. However, since these bacteria are not indicator of viral or protozoan contamination, and although they are used as an indicators for fecal pollution in the environment, they don't necessarily provide a good indication of pathogen reduction in fecal sludge treatment process. Total and fecal coliform are not good indicators in tropical and subtropical climates. The standard method for coliform analysis relies on the production of acid and gas in the medium incubated at the temperature equal to the temperature of human body. The standard method of analyzing thermal tolerance in fecal coliform relies on the production of acid and gas from lactose when incubated at 44 degrees Celsius. However, under tropical and subtropical conditions, it has been found that coliform, which are not necessarily fecal, also grow and produce acid and gas from lactose at 44 degrees Celsius. Enzymatic and biochemical assay have been developed for the detection of E. coli, and commercial kits are also available for total and fecal coliform and E. coli. When analyzing fecal sludge, helminths are most commonly used as an indicator for effectiveness of pathogen reduction due to their prevalence in low and middle income countries and their persistence following treatment. Helminths are eukaryote parasites worms which are prevalent in about 1/3 of the world's population. Helminths include nematodes that are roundworms, cystodes, flatworms, and trematodes that are flukes. These are important pathogens to monitor as eggs from these infected persons can affect hundreds of people. Ascaris lumbricoides, a type of roundworm, is the most commonly used indicator as the eggs are one of the pathogens most resistant to inactivation in treatment process, can be identified relatively easily. Another class is viruses. Quantification of total viruses in fecal sludge can be undertaken using an electron microscope, but the easiest method of evaluation is to measure their efforts on hosts. The bacteriophages are commonly used as viral indicators. Thank you so much.